Um, so I hope you can hear my voice and my voice is clear. Please let me know in the chat box that you can hear me, and also let me know in the chat box that you can see the screen. Okay, I will initiate the slideshow, and anytime there is any kind of problem, please let me know. So today we're starting off with a new topic, that is isolation and screening of industrially important microorganisms. So in this chapter or topic, we're going to look into different kind of methods that are used to screen those kind of microorganisms which are useful for fermentation, and those kind of microorganisms which are beneficial. So we have seen in introductory chapters and in media and so much more that there has to be a microorganism, and then you have to provide the nutrients, you have to provide the media for that particular organism. That all stands true, but the thing is, where do you get that organism, and how do you select that kind of organism, right? So for those kind of organisms, you have to do some sort of screening, and this chapter and topic discusses about what kind of screening we can do to get some useful bacteria or any other microorganism which can be used later on industrially to give us some benefits. They could be pharmaceutically important microorganisms, those that are producing antibiotics, or it could be any other microorganism which is producing any kind of organic acid, or it is producing any kind of metabolites as well, including vitamins and so on. So there are different ways to screen them, and usually the screening is divided in two kind of methods, and we're going to see that shortly. But this slide is discussing about what kind of very generally, what kind of microorganism do you really want for industries? So what happens that fermentation industries, they usually prefer grass microorganism. GRAS, grass microorganism. What are these grass microorganisms? These are generally regarded as safe, right? So G for generally regarded as safe. It's an acronym, right? So grass microorganism, because why they're generally regarded as safe, they shouldn't be pathogenic bacteria. You can't culture pathogenic bacteria in any fermentation industry or any food industry in which you want to obtain some sort of product, because that is not safe. You can't actually culture a pathogenic, harmful bacteria, right? So this generally regarded as safe microorganism are those which are non-pathogenic bacteria and are not known to cause any kind of disease. Moreover, in fact, they are beneficial because they have some sort of good properties and traits which are beneficial for human consumption or elsewhere, right? So, um, but the thing is, there is requirement for process and product approval using a new microorganism for stringent and associated costs are much higher, right? So why aren't we not restricted to just few microorganisms that we know they are safe? Why are we in search of new microorganisms? Because we want to modify our products, and we want to get new products, and new products which are somehow cheaper for us, because if we go for synthetic synthesis, it usually is not useful, and people they consider products coming from natural sources easy. So I mean, no matter what kind of justification you give to this process, it's never going to end. Science and research and industries are always going to search for new kind of microorganism, so for better properties, maybe for automation of the process, to improve the process, anything. Okay, so isolation of suitable microorganism from the environment. So basically what happens, how do you find something entirely new? Okay, there are microorganisms that you know already, that this microorganism is known for producing this kind of property, antibiotic, anything. But you're always in search of a new kind of microorganism, and you search for new kind of microorganism from the environment. And, and what kind of basically approaches or strategies are there to get a new microorganism from the environment? There are two types. You would go for a shotgun approach, or you would go for an objective approach, right? So what happens in a shotgun approach? In shotgun approach, you go for microorganism and samples from different areas. You can collect it from animals, you can collect it from plant material, soil, sewage, waste stream, from wherever you want, and particularly from unusual habitats. And then what do? You isolate them and then screen them for desirable traits. This is shotgun approach. So you have no objective laid out whether you want a bacteria that is producing an antibiotic, because that finding the antibiotic producing bacteria is an objective. But in shotgun approach, you do not have that objective, and you would randomly select samples from the environment, and then you would isolate and screen them for any kind of trait. What happens in objective approach? That for objective approach, you define the sampling site, right? So you go for sampling from specific sites where microorganisms with the desired characteristic are considered to be likely components of the natural microflora. And how come? Let's just say that you want any microorganism that is actually clearing out the oil and the petroleum and hydrocarbon degrading microorganism, right? So from where are you going to isolate such kind of microorganism? Of course, you are going to isolate the microorganism from areas which have been exposed to oil spillage. And after you take that spillage soil and evaluate and try to find out the you screen and then you isolate, and you come up with microorganisms on your culture plate. The kind of microorganisms you would have are likely to be those which can survive on higher concentration of hydrocarbon, and they can survive on higher concentration of hydrocarbon because they have the ability to utilize that hydrocarbon. So now you have an objective in mind and you have a specific site. But there's an example written over here, and let's look into that example. So what it says, when attempting to isolate a microorganism that can degrade or detoxify a specific target compound, sites may be sampled that are known to be contaminated by that material. Like I just mentioned, that if you want a bacteria that can degrade hydrocarbon, then you go to spillage sites that has petroleum spillage in a few years time, or actually less than few years time. You know that the soil has been contaminated by petroleum spillage. You would go collect that soil and use that as a sample to screen the microorganism. Now, what happens once you have done the sample collection? What do you do next, right? So the initial step is often to kill or repress the proliferation of common organism and encourage the growth of the rare ones, right? So what happens that once you have an environmental sample and you make it and you make you bring the sample in the lab, then you place the sample on the nutrient media or any other nutritious media that you have, and you let the growth of microorganisms take place. It's not that you're only going to get your particular desired kind of microorganism. You're going to get dozens and dozens and thousands of colonies, hundreds of colonies, and most of them would be very common microorganisms which are usually not desired. So what you're supposed to do? You're supposed to eliminate those common microorganisms, and you want to only extract, screen, and isolate those which are rare ones. So you have to adopt methodology in which the common microorganisms are suppressed, and those which are rare and desired, they are encouraged to grow in that environment. So how many screening techniques are there? They're actually they're numerous. Actually, this section is, it's too big, right? How many screening techniques? There are many multiple screening techniques, and it depends upon your objective primarily, right? But if you were to divide in any categories, you would divide it in two broad categories, and that would be primary screening and secondary screening. So the two categories are simple, primary, and secondary. What happens in primary screening of microorganisms? They may be defined as detection and isolation of desired microorganism based on qualitative ability to produce a desired product like antibiotic, any kind of amino acid, or an enzyme. So, so basically what you want to do, you want to get a sample, and then you want to isolate a microorganism just by, uh, there's a qualitative assessment. You're not doing a quantitative assessment, or you're not doing a more detailed assessment. So primary screening is just to get a desired microorganism on your culture plate. But secondary screening, what happens that you go in much detail. You try to find out what kind of, if you were trying to identify in primary screening any antibiotic producing bacterial strain. So what would happen that in the end of primary screening, you would have a culture plate with a colony growth that you know that this bacterial colony is producing an antibiotic. But it's an antibiotic against which kind of strain, and what is the chemical structure of that antibiotic, or what other chemical details you do not have in primary screening method. So what do you want? You would move on to a secondary screening methodology in which you're going to get more, you will do more research, and you will get more details about how you can how to understand the products more. If it was antibiotic, you would want to know this antibiotic works against how many strain, what is the MIC of this antibiotic, what is the minimum inhibitory concentration, and what is the dose, can it be processed, can it be manufactured. So there are so many details, but you would do them once you have done the primary screening. So usually it is desired for microorganism to be isolated from a much larger population of different species of microorganism. So this is what primary screening would do. It would give you microorganism from a large population to, it will narrow you down to very few microorganisms which are actually desired. So let's discuss some of the important primary screening techniques, which would give you a very good idea. These five will actually give you a very good idea of how these techniques are working. So there are five that we're discussing: the crowded plate technique, indicator dye technique, enrichment culture technique, oxanographic technique, or technique of supplementing volatile and organic substrates. So what's the crowded plate technique? It is very simple. You must have already done that. You must have already heard of this. And then there are a lot of variation in a crowded plate technique. So what is a crowded plate technique? You bring in a sample, environmental sample, and most commonly what happens that the environmental sample is the soil. So let's take soil an example and continue further. So you're taking soil and you're making serial dilutions out of this soil. What is serial dilution? That you're going to consecutively dilute a single sample until you have many dilutions of the sample, initial sample. And what you're going to do, you're going to place the serial dilutions, each one of them, on the plate And you're gonna repeat the process until you get 300 to 400 individual colonies on a single agar plate, right? And once you have this 300 and 400, this number is important because this number makes this process a crowded plates technique, right? Because the number of colonies that are there on the plate and which are being assessed are too many, and 300 and 400 tiny spots of colonies growing on a petri plate is actually a lot, right? That's a very large population. So the antibiotic producing activity of the colony is indicated by no growth of any other bacterial colonies in vicinity. So this region of no growth is indicated by the formation of a clear, colorless area around the antibiotic producing microorganism. So let's say that this soil sample was there to identify bacteria that are producing antibiotic. So what happens that let's say if I'm going to draw, and let's see if we are successful in actually coming up with a picture. So let me just draw. The circles that I've drawn here is basically the culture plate, right? Mm. Oh, this is what I did not want to do, but let's see like this. Right, so this is your culture plate, and you are growing. There is agar on this culture plate, right? Next, what do you want to do? You want to see that there are bacterial growth, and that bacterial growth over here is in green color. All right, so this green color spots multiple bacteria which are grown here, right? So it's a crowded plate. It's a very crowded plate. So what happens that since you want to identify among from these colonies the kind of bacteria which is producing an antibiotic. So imagine that this bacteria over here, let me point it out. This one here, right? And I'm gonna call this as A, right? A, and let's pinpoint this A1. This A colony here is a colony which is producing antibiotics, right? And since this colony is producing antibiotics, there should not be any growth of bacteria around it, right? So, so there is multiple bacteria growing of different types, which are growing over here. Okay? So it's so congested that it's crowded. But what happens that the area around this bacteria, particular this one, there, right? So imagine that your plate is so crowded that oh my god. So what do you see here? What you see is that the bacteria, it has a very clear zone around here. So you can see that this a microorganism is not letting any other bacteria to grow around it. Why? Because it is a kind of bacteria that is producing an antibiotic, right? So since it's producing an antibiotic, and so, so after you follow this procedure and you get this growth inhibitory zone around your microorganism, you know that this is your desired bacteria. And you will not see the same kind of zone formation anywhere else on the plate. You will only see it here. I hope my point over here is clear. Okay. So what happens that there are certain side effects, not kind of side effects, but downsides of this crowded plate technique. The downside is that you do not know against what kind of strains this antibiotic works. So in order to take care of this kind of phenomena, what do? You use the test organism, and you use test organism so that you can see what is this test organism, a known microorganism against, and you want to see whether this antibiotic work against that bacteria. For example, your test organism, I'm pointing it out here, that your test organism could be E. coli, right? And why E. coli? Because you want to see that the antibiotic that A is producing, does it work against E. coli or not? So E. coli, what is E. coli in, as an example, would be a test organism. All right. So what you can do in order to test whether the antibiotic is against E. coli, what you can do, what you can modify your crowded plate technique, and in this modification you're only going to use 30 to 300 on the plate, and once you have achieved these colonies, you're going to flood the petri plate with the test organism, and you're going to let test organism grow. And if you see the zone of inhibition against a colony, that means that colony is producing the antibiotic which is effective against the E. coli. And this is a modification of the crowded plates technique. Then let's look into indicator dye technique. What happens in indicator dye technique? That in this you can use a dye. You can incorporate a pH indicator dye, and once you incorporate this pH indicator dye in the nutrient medium, there will be change of color, and change of color which show that this colony is producing certain kind of compound. Since you're using pH indicator, so what kind of compounds are you targeting? You're either targeting acids or you are targeting amines, right? Amines are basic in nature, acids are acidic in nature. So if you want to see whether a colony, a bacteria colony, can produce an organic acid, and you have used a pH indicator, it could be neutral red, it could be bromo, thymol, blue, whatever, or it could be other pH indicators. So what it will show you? It will show the color change, and the change will be more on acidic side. So you know that the organic acid has been produced, and this bacteria is the one which is producing the organic acid in the vicinity of which there is a color change. Vicinity means closely, right? So if there's a bacteria and there's a color change around that bacteria, only that colony, then that means that bacteria is producing that organic acid. And it can go the opposite side. If you're not looking for the acid, you're looking for amines or any basic compound, the color change would be towards the basic pH. And you would know right away that the bacteria is producing this kind of microorganism, right? Sorry, this kind of compound, a basic compound. Then there is an alternative to this method, and that alternative is that you can use calcium carbonate. Calcium carbonate is a salt, and calcium carbonate is white in color, right? Calcium carbonate is white in color. So when you're going to add calcium carbonate in the agar medium, it's going to make the medium cloudy. All right. So there is going to be white cloudy medium. And then you're going to let microorganisms which can grow organic acid to grow on that nutrient media. Then you're going to see clear zones. So once you're going to see clear zones around the colonies, it means that calcium carbonate has been converted to. Okay, so where were we? There was a small distortion. So what happens that once you've added calcium carbonate and you've added calcium carbonate in the medium, it's going to make the medium cloudy. And once you grow a bacteria which is producing an organic acid, it's going to react with calcium carbonate and it's going to convert calcium carbonate in a salt in a salt version. Like it's an example that your calcium carbonate, when it reacts with an acid, for example HCl, it will convert your calcium carbonate to calcium chloride, which is common salt. So calcium chloride, when dissolved in agar medium, it's colorless, right? So whenever this calcium carbonate is going to react with any kind of organic acid, it's going to convert calcium carbonate to a salt version, which is colorless. So calcium carbonate, when it was added, it was whitish and cloudy. So the bacteria which is producing organic acid, the area around that media is going to be a clear zone, right? So it is yet again another indication. Then what do you do? The identified colonies can be streaked again and again on other plates, and a stock culture can be maintained so that you can use them further. Then enrichment culture technique is very simple. Enrichment means you add more and more nutrients. So we do know that every bacteria and every group of bacteria they can differ in terms of their requirement. So if you're using a commonly used nutrient media or commercially prepared media, and you're not going to get any kind of exotic or a new microorganism. So if you want to really identify a new kind of microorganism, you would go for enriching the culture technique with new kind of nutrients, substances, chemical compounds, vitamins, or any such thing. You can add it as an extra nutrition or an extra enrichment within the media so that you can get a new variety of microorganisms to grow. So enrichment technique is something very simple. You modify and you improve and you enrich the media already in hand. Then there is this method in which you identify the auxotrophs. What are auxotrophs? Auxotrophs are those kind of bacteria uh, which require any particular um, nutrient for its growth. So I'm just using an example, just a hypothetical example to defend this definition. Let's say there is a bacteria A, and bacteria A can only grow if it's provided with vitamin A. If vitamin A is not in the media or not in any kind of source on which it's growing, on the growth requirement, right? So if it doesn't has in the media vitamin A, then an auxotroph bacteria A cannot grow in that media. But if the media has vitamin A, then an auxotroph can grow. So what is an auxotroph? It requires that if a particular kind of a specific kind of compound as a requirement. If that particular compound is missing, then this kind of bacteria cannot grow. So what is happening that we are trying to identify that this technique is employed for detection and isolation of microorganisms capable of producing extracellular substances such as growth stimulating factors like amino acids, vitamin C's, or any vitamins, etc. Right? So you can use this to find bacteria which are able to give you that element, right? So in this example, we've just said that a bacteria requires vitamin A. So if you use a bacteria and you use sample provided colonies and you let this bacteria to grow, you're going to see that only a single type of organism will be able to grow, and that would mean that it is producing that vitamin, right? Right. So let me read. This is the procedure. The second point here. I'm going to read and I'm going to draw so that my point is clear. So for this purpose, spread a suitable aliquot on the surface of the sterilized agar plate and allow the growth of isolated colonies after incubation. A suspension of test organism with growth requirement for a particular metabolite is plotted on the above plate containing isolated colonies which are subjected to further incubation. So let me draw this and it's going to be inshallah very clear to you, right? So it's a method similar to crowded plate method, it's not much different. So here is a Petri plate and you are growing, let's say, colonies, which are again green in color. So these are few colonies which are on the plate, which are growing once you've added the environmental sample. Once you've added that aliquot, this aliquot is that sample, that dilution from the environmental sample, right? And now what happens that you are going to add a suspension of the test organism with growth requirement for a particular metabolite, right? So let me draw first a test organism, right? So I'm going to draw a test organism. So there is this flask, right? Right? This flask contains a broth culture and it contains a broth culture of a test organism and that test organism is A, right? So this A is an auxotroph which has a requirement of vitamin A. All right. So A will not grow until it gets vitamin A from the media, right? So, and this media, this agar plate, it doesn't has vitamin A. All right. So what do you do? You add a suspension of the test organism, and we have the test organism A, and you have added the suspension of test organism, right? So you are pouring it here, and once you pour it here, let's say you have poured it all here. ठीक है यू हैव आपने अपना टेस्ट ऑर्गेनिज्म इस पेट्री प्लेट के ऊपर उसकी सस्पेंशन डाल दी ठीक है जी अब होगा क्या कि विटामिन ए दिस बैक्टीरिया ए इसको चाहिए विटामिन ए ठीक है जी और विटामिन ए इस मीडिया की कल्चर प्लेट के अंदर विटामिन ए नहीं है बल्कि होना ये चाहिए कि एनी ऑफ द बैक्टीरिया जो कॉलोनी यहाँ ग्रो हुई है ये कॉलोनी 1 2 3 4 5 इनमें से कोई एक कॉलोनी ऐसी कॉलोनी होगी जो कि विटामिन ए प्रोड्यूस करती होगी ठीक है तो बैक्टीरिया जो है ये वाला पिंक जो इधर ड्रॉ किया है टेस्ट ऑर्गेनिज्म ये वहां ग्रो करेगा जहां कॉलोनी विटामिन ए डेवलप कर रही होगी ठीक है जी so let me further modify this point and let me say that this colony here this one is the one which is producing vitamin A ठीक है vitamin A produced by colony 1 right colony 1 is producing vitamin A तो होना क्या चाहिए what should happen that only this colony is going to grow like this, right? After incubation, आपका टेस्ट ऑर्गेनिज्म बाकी कहीं भी सरवाइव नहीं करता, आपका टेस्ट ऑर्गेनिज्म सिर्फ इसके अराउंड सरवाइव करता है. Why? Because this test organism requires vitamin A, and vitamin A is being provided by C1. So what happens that this and this are going to grow together because this C1 provides the required uh, required nutrients. Okay, the rest of the process is simple. What do? You culture this once you have obtained your C1. C1 is your desired colony. So you pick up C1 and then you maintain the C1 further on, and you see whether it gives you a, you further streak it down and you maintain the stock so that you can move the colony and the technique towards secondary techniques, right? So technique for supplementing volatile organic substances. What is this technique? This technique is employed for detection and isolation of microorganisms capable of utilizing carbon source from volatile substrates like hydrocarbons, low molecular weight alcohols, or similar carbon sources, right? So what kind of compounds are you targeting? What the bacteria which is producing volatile compounds, right? So I mean, bacteria which is capable of utilizing the volatile compound. So suitable dilution of a microbial source like soil suspension are spread on the surface of the sterile agar medium containing all the nutrients except one mentioned above. And what is that? Alcohols and uh, hydrocarbons or any other which is volatile. The required volatile substance is applied on the lid of the Petri plates, which are incubated by placing them on an inverted position. Although enough vapors from the volatile substrates are going to spread on the surface of the agar closed atmosphere, and only that bacteria would grow that is able to utilize the compound. So in order to I'm going to follow the same procedure. I'm going to draw it down so it is a little more clear. So what do we have here? We have a Petri plate, all right, and and we have the lid of the Petri plate, right? So what is this? This is the lid, right? And what is this? This is the base of Petri plate, right? So now what would happen that you would take your sample and you're going to let your sample spread here, right? So you're going to spread the sample and you're going to spread the sample like here in a very light color. You spread it on your plate here, right? So this is your sample, test sample on the Petri plate here. And on the lid, what you're going to do, you are going to place a volatile compound like hydrocarbons or low molecular weight alcohols. So let's use volatile compound as this. This is the volatile compound, right? Let's say this is the volatile volatile hydrocarbon. Right? What is this? Volatile hydrocarbon, right? So this is volatile hydrocarbon. So now what you're going to do, you're going to place the lid, cover the Petri plate, and going to rest the Petri plate in an inverted position. Or and you people already know कि Petri plate को inverted position में usually हम रखते हैं, right? So what you're going to do next, you're going to plate the Petri plate. Oh, there's something not right with the way I'm drawing. So let's say this is the Petri plate, like this, and this is the cover of the Petri plate, and you have placed the Petri plate in an inverted position, right? But do keep in mind that this is the same plate, so there is a volatile compound here on the lid, right? Like it was there. And then there is sample which was placed on the Petri plate, so there is sample here. It's the same plate now placed inverted, right? So what's going to happen now that this volatile compound, it is converted into vapors. ठीक है ना? Volatile compound क्या होता है जो evaporate हो जाता है. तो ये compound आपने lid पे लगाया है लेकिन वो evaporate होगा. Evaporate होके वो कॉलोनी ग्रो करनी चाहिए जो कि volatile या evaporated गैस वेपर को यूज कर रहा है, यूटिलाइज कर रहा है एज अ सोर्स ऑफ न्यूट्रिएंट or any kind of a nutrient which is required by this bacteria. तो यहाँ भी कौन सा बैक्टीरिया होगा, एक ऑक्सोट्रोफ बैक्टीरिया होगा जिसको एक अल्कोहल की या हाइड्रोकार्बन की जरूरत है और जो कि वॉलेटाइल कंपाउंड्स की उसको जरूरत है. तो आफ्टर सम टाइम when you're going to incubate, what you're going to get, you are going to get a few colonies which are probably growing. ठीक है जी, और वो कॉलोनी क्या हो गई, एक कॉलोनी इधर, एक कॉलोनी इधर, बस दो ही कॉलोनी ग्रो कर रही हैं. एंड विच आर दीज टू कॉलोनीज, these two colonies are the ones which are utilizing the volatile hydro compound. So I hope this is clear to you all. Then we come to secondary screening of microorganisms. But we're going to do secondary screening of microorganism inshallah in the next session. So for now I'm ending this session.